Good morning, students. Today's lecture focuses on practices for collection of milk at farm level, which forms the foundation of clean milk production. As dairy technologists, you must understand that milk quality is not determined at the processing plant but at the point of production. In this session, we will systematically examine hygienic practices, milking procedures, handling protocols, and preliminary quality control. Let us begin with the fundamental principles of hygienic milk collection. Milk must be safe. This implies absence of pathogenic microorganisms and contaminants. Milk must be clean. Strict hygienic standards must be maintained during milking. Milk must be kept cold. Rapid chilling is essential to suppress bacterial multiplication. Milk must be handled quickly. Time control is critical because microbial growth is exponential. These four principles, safe, clean, cold, and quick, constitute the scientific basis of hygienic milk collection. Clean milking practices directly influence milk quality parameters. First, superior milk grade is achieved when contamination is minimized. Second, shelf life increases. Lower microbial load delays spoilage. Third, somatic cell count is reduced. Low SCC reflects proper udder health and hygienic milking. Fourth, food safety is ensured. Consumer protection begins at the farm. Therefore, clean milking is both a quality and safety requirement. Pre-milking preparation is a critical control point. The milking area must be clean and dust-free. Environmental contamination contributes significantly to initial bacterial load. The animal must be calm. Stress interferes with oxytocin release and milk letdown. Udder washing removes dirt and microorganisms. Teat drying is equally important. Moisture facilitates bacterial entry into milk. Each of these steps reduces contamination at the source. Personal hygiene of the milker cannot be overlooked. Hands must be thoroughly washed before and after milking. Use of clean gloves minimizes direct contamination. Nails must be trimmed and clean. They can harbor pathogenic bacteria. Jewelry must be removed. Such items trap dirt and compromise hygiene. Remember, the milker is a potential source of contamination. Utensil sanitation follows a standardized cleaning cycle. Step 1. Wash using warm water and approved detergent. Step 2. Rinse thoroughly to remove chemical residues. Step 3. Apply sanitizing solution at recommended concentration. Step 4. Air dry utensils. Cloth wiping can reintroduce contaminants. This wash, rinse, sanitize, air dry protocol is a standard hygiene practice in dairy operations. During milking, strict procedural control is required. Force stripping must be performed. This removes high bacterial load milk from the teat canal and helps detect mastitis. Teats must remain dry during milking. Milk must be collected in covered containers. Proper milking technique reduces physical and microbial contamination. Avoid practices such as open bucket milking, wet teat contact, or careless handling, as these compromise milk safety. Post-milking handling is equally critical. Milk must be covered immediately to prevent environmental contamination. Filtration removes visible dirt and debris. Rapid chilling to 4 degrees Celsius is the most effective method to inhibit microbial growth. Timely transportation ensures that the cold chain is maintained. Failure at this stage results in irreversible quality loss. Preliminary quality checks must be performed before dispatch. Milk should be filtered using stainless steel strainers. Visual examination must confirm normal color and absence of clots. Odor assessment should detect any abnormal smell. Temperature must be recorded immediately. This confirms whether chilling requirements are met. These checks act as farm-level quality assurance measures. Good collection practices have measurable outcomes. They significantly reduce bacterial load. They extend shelf life. They improve sensory characteristics such as flavor. They reduce rejection rates at the dairy plant. In other words, farm-level hygiene determines industrial efficiency. Let us summarize the complete sequence. Preparation involves clean surroundings, calm animals, and proper udder hygiene. Milking requires clean hands, force stripping, and contamination control. Handling includes covering, filtering, and chilling to 4 degrees Celsius. Quality check involves visual inspection, odor assessment, and temperature recording. This integrated approach ensures clean milk production. To conclude, milk collection practices at the farm level form the first critical control point in the dairy value chain. If hygiene is compromised here, no matter of processing can fully restore quality. As future dairy technologists, you must prioritize scientific milking and handling practices. Thank you.